welcome to Axonovyn. This is the place where we dive deep into the fascinating world of the neurological diseases and explore their cutting edge treatments. Whether you're a healthcare professional, a curious learner, or someone seeking to better understand these conditions, you're in the right spot. At Axonovyn, we believe in making complex science simple, engaging, and inspiring. From the latest research breakthrough to practical insights, we're here to empower you with knowledge and spark curiosity about the brain and beyond. So, hit the subscribe button and start your journeys into the incredible world of the brain with us. First of all, what is multiple sclerosis? Multiple sclerosis, or MS, is a condition that causes inflammation in the central nervous system, or CNS, impacting around 1 in 200 women and slightly fewer men in the US. The name multiple sclerosis reflects the disease's features: numerous damaged areas in the brain and spinal cord that lead to a variety of neurological symptoms over time. These damaged regions, known as plaques or scars, are a hallmark of the condition. Although MS was identified nearly 140 years ago, its impact cause remains unknown, and the definitive cure has yet to be found. However, significant progress has been made in managing the diseases, alleviating its complications, and improving the overall quality of life for those living with MS. Let's go to etiology. The etiology section briefly states that the cause of MS is unknown, but it is believed to result from a combination of genetic susceptibility, environmental factors, and immune system dysfunction. Various triggers, such as viral infection, particularly Epstein-Barr virus or EBV, are associated with an increased risk of MS. Additionally, vitamin D deficiency, geographic location, and other environmental exposures are considered contributing factors. Genetic predisposition, including specific HLA genes, also plays a key role in determining susceptibility of MS. Now, I want to talk about pathophysiology. The primary pathological process in MS is the loss of myelin surrounding CNS axons. This demyelination disrupts nerve conduction, leading to functional impairment. It is accompanied by axonal damage and inflammation, involving immune cells such as lymphocytes, macrophages, and antibodies. The demyelination process can cause severe neurological deficits and is often visualized as lesion on T2-weighted MRI scans. The symptoms of MS depend on the location and size of these lesions, particularly in areas like the periventricular, white matter, spinal cord, and cortical regions. Early in the disease, lesions are inflammatory, while in later stages, chronic lesions show less inflammation but greater axonal loss and damage. On T1-weighted MRI scans, chronic lesions may appear as black holes, which signify areas of irreversible axonal loss and severe tissue destruction. These black holes are linked to long-term disability progression in MS patients. The behavior of T-cell subsets is crucial. Certain subsets of T cells, such as Th1 or T helper 1 cells, produce pro-inflammatory cytokines like interferon gamma and interleukin 17, driving inflammation. Conversely, regulatory T cells or Tregs, which suppress immune responses, are reduced or dysfunctional in MS, particularly in progressive forms of the disease. This imbalance between Th1, Th2 cells and Tregs further exacerbates inflammation and tissue damage. In progressive MS, inflammation may play a smaller role, while neurodegeneration becomes the dominant process. Damage to axons and myelin are continuous, leading to loss of function even without significant immune cell activity. Certain pathways, like the production of TGF-beta and other mRNA markers, are dysregulated in progressive stages. Finally, while the specific triggers for this immune attack remain unclear, this immune response is highly complex, involving antibodies, cytokines, and a wide variety of molecular signals that are yet to be fully understood. Figure shows the pathology of MS. Here you can see the peripheral immune activation. In the periphery, antigen-presenting cells or APCs such as dendritic cells, macrophages, and B cells present myelin antigens to naive CD4 positive T helper cells through MHC. MHC is major histocompatibility complex molecules. Here you see the co-stimulatory signals like co-stim are required to activate these T cells. T cell differentiation. Activated CD4 positive T cells differentiate into specific subsets like Th1 cells and Th17 cells. Th1 cells secrete interferon gamma, that is a pro-inflammatory cytokine and promoting inflammation. CD17 cells secrete interleukin 17 that enhancing immune cell recruitment. Here you can see crossing the blood-brain barrier. Activated T cells express adhesion molecules like VLA4 that interact with VCAM1 and the endothelial cells of the blood-brain barrier or BBB. Matrix metalloproteinases or MMP are produced which degrade the barrier, allowing immune cells to enter the CNS. Here you can see immune process in the CNS. Once inside the CNS, activated immune cells attack myelin and oligodendrocytes. CD4 positive T helper cells interact with myelin antigens, releasing cytokines like interferon gamma from Th1 or T helper 1, promotes inflammation. Interleukin 17 and interleukin 23 from Th17 recruit more immune cells. CD8 positive T cells directly kill oligodendrocytes and damage axons. B cells and plasma cells produce antibodies against myelin components, facilitating further destruction. And macrophages engulf myelin debris and release reactivate oxygen species, contributing to tissue damage. Here you can see oligodendrocyte and axonal damage. These cells produce myelin to insulate axons. In MS, they are destroyed by immune responses, impairing myelin repair. Oligodendrocyte progenitor cells, or OPCs, attempt to replace damaged oligodendrocytes and repair myelin. However, this process is disrupted due to the chronic inflammatory environment. LINGO-1 is a protein that inhibits remyelination. Blocking LINGO-1 has been explored as a strategy to promote myelin repair. Here you can see the demyelination and axonal damage. Normal versus demyelinated axons. Normal axons are insulated by myelin, ensuring fast and efficient signal conduction. Demyelinating axons lost this insulation, impairing nerve conduction. To compensate, sodium channels appear along demyelinated axons, but this process is energy intensive and ultimately unsustainable, leading to further damage. Chronic demyelination results in irreversible axonal transection, contributing to permanent neurological deficits and disease progression. Here you can see the regulatory mechanisms. Tregs, or regulatory T cells, produce anti-inflammatory cytokines like interleukin 10 and TGF-beta to suppress immune activity. In MS, the balance between pro-inflammatory like Th1, Th17 and anti-inflammatory like Tregs is disrupted, leading to unchecked immune attacks on myelin.